Very good evening. Today we discuss on a topic RNA interference, which is related to the biotechnology. Here, what is the RNA interference is? A host double-stranded RNA. It targets mRNA of pathogen and no protein synthesis takes place. This is the mechanism is related to the RNA interference. Here, when the pathogen, when it enters into the host cell, when the pathogen enters into the host cell, this way, and this pathogen is interacting with the genetic material of a host and it is going to produce enormous number of RNA, the single-stranded RNA, and it is going to produce n number of proteins. It is encoding n number of proteins, those proteins which kills or may cause disease in the host cell. Here, this pathogen, when it is producing the single-stranded mRNA and it is going to produce the proteins, this is translation level actually. Translation level. As you people knows that the CD, central dogma, it consists of replication and transcription and translation. And this is the out of topic of this RNA interference. Anyway, here in the translation level, this pathogen is producing proteins from single-stranded RNA. Here, pathogen enters into the host cell and it is producing mRNA in the host cell with the help of the genetic material of host cell itself, and it is encoding n number of proteins. These proteins may kill or may cause the disease in the host cell. To stop this mechanism, the double in any means of the host cell produces double-stranded RNA and it targets mRNA so that no protein synthesis takes place. Here, how this mechanism takes place, as I shown here, it will not complete in these three steps simply. If you see here, double-stranded RNA, if it is produced by the host cell, what will happen is the dicer, dicer is the restriction endonuclease 3 type of enzyme, whereas the restriction endonuclease, it cuts the genetic material in the middle. Here. So dicer, which is already present in the host cell, this dicer cuts this double-stranded RNA into small fragments. What we call these fragments are small interfering RNA. These fragments complexes with ribonuclease components. So these small interfering RNA complexes ribonuclease components, and we call the component of this complex, we call the risk. What is the risk is? The risk is here actually the RNA inducing silencing complex. That is what the risk. Here, when the dicer cuts this double-stranded RNA into small interfering RNA, it forms inactivated risk actually. Why it is inactivated risk is it's already double-stranded, so that it cannot bind with another genetic material or like RNA, single-stranded either DNA or RNA, because already it is in double-stranded, so that it is inactivated. When it unwinds, when it becomes a single-stranded, it's unwind, it become single-stranded. Now it is called as active risk. Why it is active risk is it is a single-stranded. There is a chance of binding with some other DNA, single-stranded DNA or single-stranded RNA, because it is single-stranded here. So here, this activated risk, it is going to bind with the mRNA of pathogen. This is what the green color, the mRNA of pathogen, it is binding, and what will happen is when it binds, already the dicer which is present already in this component or in the complex, it cuts or when it is binding, this mRNA fails to produce the proteins. As I said here, it is a translation level actually. In the translation level, this pathogenic mRNA, if it is individual, when it is individual, it is going to produce proteins. As I said, these proteins may kills or may cause disease in the host cell. So, not to leave this mRNA alone, the pathogenic mRNA not to live in as alone, this risk, activated risk is binding with this mRNA so that the translation will not take place. So no protein synthesis takes place, no proteins produced in the host cell, so that the cell remains healthy. Here, what is the risk here is, the risk is the, as I said here, the risk, it is a ribonuclease complex actually. What are the components present in the risk is the dicer, the slicer and argonaute. These are the three. Here, the double-stranded RNA, which means produced by the host cell, it is capable of producing three types of mRNA, siRNA, shRNA, mRNA, with the help of dicer reactions. The dicer, it cuts double-stranded DNA into three types of fragments. These are the siRNA, what it is small interfering RNA, and shRNA, small hairpin RNA, miRNA, it is microRNA simply. In this module, we discussed only on siRNA, small interfering RNA. So this is what the mechanism, how the pathogenic single-stranded mRNA is arrested by a sense of double-stranded RNA with the help of different type of risk or like a dicer, different type of proteins here. In the upcoming videos, we discuss on some other like shRNA, miRNA. Thank you.